Hello, everyone, and welcome to the Relapsed Refractory Myeloma Patients chapter brought to you by the Myeloma Crowd Community Program by HealthTree. My name is Audrey Burton-Bethke, and I am the Myeloma Crowd Community Manager. I'm going to share some slides to share a couple of announcements with you, and we can get started. So, today we are going to be hearing from Dr. Nina Shah, a multiple myeloma specialist who will share with us more about the Abecma ide-cel CAR T Therapy. She will be introduced formally in just a moment by Jenny Ahlstrom, the founder of Myeloma Crowd by HealthTree. Jenny will be moderating today's event. I'm grateful that you're here. It's fun to see people joining us, and we're excited to hear about an important topic. Today's session will be recorded and will be sent out to all registrants 24 to 48 hours after this event has concluded. We'd like to thank our sponsors, AbbVie, Bristol Myers Squibb, Amgen Oncology, Karyopharm Therapeutics, Genentech, Oncopeptides, Adaptive Biotechnologies, and Takeda Oncology. So for today's topic, as I mentioned, it will be Know Your Myeloma Therapy, the Abecma ide-cel CAR T Therapy. And I'm going to turn the time over to Jenny to discuss why we chose this topic for this chapter, and then she will formally introduce Dr. Shah. So Jenny, the time is now yours.
Well, welcome everybody, and thanks for joining us. This topic is a really important one because it's really groundbreaking. It is the very first CAR T therapy that's been approved in multiple myeloma care. And so it represents an opening of a door, in my opinion, to a completely new type of therapy. It's exciting because CAR T therapy is a one and done therapy. As far as we know it, I'm sure we might have maintenance therapy be added a little bit later as we learn more. But to open this new door is just so incredible for myeloma patients. So I would like to go ahead and introduce Dr. Shah, who is going to be presenting for us today. Hi, Dr. Shah. We're so happy to have you. Dr. Nina Shah is a specialist in blood diseases who focuses on treating myeloma. So her professional interests include the intersection of this immunology and oncology, which in my opinion is a unique twist because not all myeloma specialists are specialists in the immune system as well. And we're really seeing this whole group of immunotherapies come to the clinic. So the more we can get experience like Dr. Shah's for both of these, the better they're going to be able to understand what's happening in the immune system. Dr. Shah earned a bachelor's degree in cognitive neuroscience at Harvard, followed by a medical degree from New York University School of Medicine. And now I want to know how you got involved in myeloma. She completed a residency in internal medicine at Columbia University and a fellowship in hematology oncology at MD Anderson. So, Dr. Shah, I'm going to turn the time over to you. But yes, I'm curious now how you started out in neuroscience and then moved to hematology.
Thanks, Jenny, for having me. First, I want to say I'm very privileged to be invited to any of your events because I feel like you've made such a huge impact for our patients. I really appreciate all the work that you and your team do to keep us all educated, the physicians as well. I got involved with multiple myeloma. Well, I always knew I wanted to be a physician, I really enjoyed, it's funny, I have this major in cognitive neuroscience, and what that is the connection between the mind and the brain and behavior. And what I did my senior thesis on was the effect of a religious experience on salivary immunoglobulin A levels. And I say that because I've always been interested in the intersection of mind, brain, and what we do with our immune system. So it was almost fitting that eventually I went into a field like oncology, which does have a lot of impact from the immune system and immunotherapy, and particularly multiple myeloma. In a way, it was serendipitous that I came upon this malignancy. What I love about multiple myeloma patients, all of the patients, is that, as I always say, it's a marathon disease. And what that means is that I am connected to these patients for years. And I know when their kids get married, and they know when my kids go places, and I really enjoy that. And that was part of and another element of like this cognitive neuroscience major. Like I had to learn psychology and understanding that people are different. So I really feel very fortunate that my education and training allowed me to get to this point, and hopefully that I can give back to all of the patients.
Well, you are giving back and you do so much. So I know you were very involved in the ide-cel clinical trials. And so you're just a perfect person for us to talk to today. We want this to be to have a section at the end for Q&A. So as you're watching, please add your questions to the Q&A. And then when we get to that point, we want it to be, more of a conversation. And you're just so good at answering those questions. So we just appreciate everything that you do at UCSF as a myeloma specialist. I don't know how you do what you do because I see you everywhere, and I just know that you have a busy family too, and it's just incredible what you do. So my hat's off to the amazing work that you're always doing.
Thank you.
We'll go ahead and let you share your slides, and then we'll.
You know what, Jenny? I think I was told that I don't have to prepare slides, but I can.
Oh yeah, yeah. You can just have a conversation. That's fine.
Yeah. Maybe I can give an overview of what Abecma is and what CAR T cell therapy is. Would it be a very
Yeah,
simple
yeah.
overview? Would that be
No,
helpful
yeah.
to get
We
into
would
the
love.
conversation?
We can take 30 minutes or so to you can present and discuss, and then we can
Yeah,
talk about
sure.
questions.
So we're really excited about this new approval in multiple myeloma for patients with relapsed refractory multiple myeloma. This is the first cellular therapy, and what that means is this takes your own cells and engineers them to be specifically targeting the myeloma. So that's a revolution, because what we've usually done, as you guys all know, is give you drugs and an infusion or a pill, but we've never taken something from you and made it attack your myeloma. So this is really a new step for a type of a new door for a different type of chemotherapy or type of therapy in general. The other thing that's unique about it is it's targeting a protein, BCMA, B cell maturation antigen. And what's really interesting about that protein that many of you have heard of is that it's almost uniformly expressed on all plasma cells, which is the myeloma cells, but it's not expressed on normal cells, on other normal cells. So it's a great target. You always want to sort of minimize the toxicity and maximize the efficacy. And that's how to do it with a really specific target. So these two aspects of engineered T cells plus a great target make this therapy something that we're excited about. This particular T cell therapy, and I'm going to tell you about the logistics in a minute, but just so you know sort of the stats, was FDA approved recently for patients who've received at least 4 prior lines of therapy. Now, I want to really quickly let you understand what that means, because it's hard to know. Am I a candidate? Am I a candidate? That's one of the questions we get a lot. And what this means is you've had 4 prior different lines. Now, lines is a unique piece of verbiage that us myeloma doctors use and don't agree on. So don't go on Twitter and see if you'll see all sorts of different definitions of this. What a line means is a regimen that was planned for you. So often we'll consider the VRD induction, transplant, and maintenance as just one line, even though it's 3 different things, because it was a plan that you had in the beginning. But this is a little bit up for interpretation. And I hope that we as physicians are going to work with you as patients to clearly define what the lines are, because ultimately those FDA labels lead to what the insurance companies will cover. And so it's really important for us to communicate with you, okay, you've had this therapy and then this therapy and this therapy. And actually those first 2 is one line and the second one is another line. And we have to make sure you have 4 so that we can get this covered by insurance. And that'll be something that we're all learning, but it is a true real world logistic that we have to address. So the FDA has approved this for patients with 4 prior lines of therapy who have relapsed refractory myeloma and have had exposure to our 3 major classes of drugs. So there's the proteasome inhibitors like Velcade or Kyprolis, and then there's the IMiDs like Revlimid or pomalidomide, and then there's the anti-CD38 antibodies, which include daratumumab or isatuximab. So many of you know about all of those and have had exposure to them. So you have to have had exposure in whatever sequence it was, and you have to have had 4 lines. And those people will be eligible to get this BCMA CAR T cell therapy. So why did it get approved? It got approved because there was a pivotal phase 2 trial, meaning that there was a trial that was done with enough patients that showed enough good results that allowed for the FDA to say, hey, the benefit outweighs the risk, the cost outweighs, the benefit outweighs the cost, and therefore we should offer this as something for myeloma patients. So for this particular trial, 128 patients received this therapy. I'm going to talk about the logistics of the therapy in a minute, but I'll just tell you the short of it, which is that there was an over 70% of patients responded to this. Now, I want to point out these people were very heavily pretreated. So most of them, at least 50% of them, had had at least 6 lines of prior therapy. So if you think about that, that's a lot of therapy. That's years and years of therapy. We had patients with like 12 lines, et cetera. So a lot of pretreated patients, and they responded over 70%. And actually, for the recommended dose from the FDA, the 450 million dose, those patients had a greater than 80% response rate. So you have a 4 out of 5 chance that you're going to respond. And so that's something that we usually don't see in myeloma. If you've ever seen some of the recent FDA approvals, they are approved with a response rate of around 28 to 30%. So, you expect a third of people to respond. Here you have 80% of people responding. So that actually was something significant. Now, when we talk about how long the responses lasted, those responses lasted for the entire population a little over 8 months, and what we call progression-free survival. I should say, not, there's some verbiage differences, but progression-free survival, meaning how long before all the patients progress. 50% of them, a little over 8 months, 8.7 months. But if you look at the patients who got the actual recommended dose, those patients had stable disease or had a lack of progression for about a year. And if you got into complete response, then your time that you had this was 20 months, so about a year and a half. So why I really am excited about this is that you have to understand, or we have to understand, that you guys are going through so much therapy all the time. Every month you go get your labs checked. Every month you may, or every week you may come for an infusion. You have to take a pill. This is just a one-time therapy. And so with one-time therapy, getting such long duration of response has been something that we have been sort of happy to see, because we love seeing our patients, but maybe they don't like to see us so much to come to the clinic. So this is something we're excited about from that perspective. Regarding safety, there is two major side effects that have to be talked about. The first one is called cytokine release syndrome, or CRS. And what that is it's basically like a flu-like syndrome. And if you think about it, these T cells have been tricked into thinking that your myeloma is a virus. And so they see your myeloma and try to kill it, and they react just like your T cells would if you had the flu. And that's why patients will have fever, they may have low blood pressure, they'll feel really weak and fatigued. And we watch patients for this very closely at our center in the hospital. And we know this usually happens within the first 1 or 2 days of getting the CAR T cells that have been approved. And there is an antidote for this called tocilizumab, which really sweeps up some of these proteins that cause this syndrome. And it doesn't seem to affect them badly, meaning the T cells are still active even though we treat this syndrome. So that's why we monitor patients closely after those first few days they get the CAR T. And yes, over 80% of people had that, but it was very manageable. This is what we call low grade, grade 1 or grade 2, meaning patients usually did fine, even though they had that fever. Some just got Tylenol, some got this antidote, some got just some fluids. And so we were able to control it. And if necessary, we can give steroids. And that also does not seem to affect these T cells badly, although we really try to avoid steroids if we could, because of the theoretical chance it may affect this T cells. The second thing that we get are concerned about is neurotoxicity. So that's toxicity that affects the brain or any part of the nervous system. And this usually manifests as confusion, first and foremost. And I always say the patients seem a little drunk. So, and it's like you wouldn't be able to tell unless you ask some questions. And so we do have a questionnaire that we give the patients every day to understand if they're sort of still thinking straight. And they can tell, but the caregivers are the ones that can tell the most if they're not totally on track. And what this is a little bit of inflammation there, again, because of the T cells. And in this study, 18% of patients had some kind of neurotoxicity, but again, grade 1 or grade 2. And we usually control this with steroids. Pretty manageable. The most important thing about toxicities for this type of T cell therapy, in my opinion, is that it's easily, it's clearly delineated that it's very closely observed and that you jump on it as quickly as possible so it doesn't get out of hand. So most of these patients did pretty well. Yes, there were one or two bad events, but it's 128 patients of very, very bad myeloma, and I think we were okay with that data. So that's the data that helps us to understand what this therapy is doing. I just want to again compare the things that have gotten FDA approval in this patient population until this time have been around 28 to 30% overall response rate, whereas this T cell is 70 to 80% response rate. And then the progression free survivals that have been the data for the things that have been recently approved are like 3 to 4 months. And this is somewhere between maybe 9 to 12 months, maybe 12 months closer, and then depend better if you get a complete response. So the comparison between these types of therapies, this T cell therapy and what we've already had, sure, we love those drugs that we already have and we need them, but this is something new that we can have as another tool for patients who have advanced myeloma. So that's sort of the overview of the data that led the trial that led to this approval and why we're excited about it, because we think, we always think, does the benefit outweigh the risk for these patients? We're putting them through so much. Does it make sense? And to see some of my patients go back to work or to make it to things or retire, it has been really fulfilling. I understand, and full disclosure, it's not a cure. There's still what we call, there's no plateau on those curves. So if you see these curves, you'll see them still kind of going down. And at some point, people are going to progress. We don't know when, and we wish that we knew that better. But at some point, people progress. And that's important, that we monitor patients closely with labs to see that. And it's also really important that we have something lined up. And so all of these other clinical trials that you hear about that might be, other drugs, non-BCMA drugs, those are important because they help us to give another option in this marathon disease. So that's an overview of what ide-cel is. The trade name for it is Abecma. And I'll say it again, but there is actually a website called abecma.com that you can go to. It will tell you the things I said. I don't work for BMS at all. This is purely for patient information, that you can actually go on there and caregivers can get some understanding as well. I want to talk, Jenny, if it's okay, about the logistics of what this means for
Oh
patients.
yeah,
Would that be
absolutely.
okay? Okay.
Yep.
So one of the things that's like hard to wrap your head around with this type of therapy is that it's not your usual come in every week, go to your oncologist's office, get an infusion, go home. It's not. And that's a good thing and that's a bad thing because it's sort of like transplant. There's a lot of upfront investment on your part. So what this means is when this is available and your doctor says, okay, you're ready to go to CAR T, what will happen is kind of similar to transplant. We'll get a few tests like an echocardiogram, some labs, et cetera. And then, of course, we're going to submit for authorization to your insurance company. That's like on us. Like, you don't have to deal with that. We'll deal with that. But then what it means for you is that you will eventually come to the center. So you have to do this at the CAR T specialized center. So for us, it's us and Stanford in our area and UC Davis. And you'll see that there are various specialty centers that have been approved to do this. So this will be, you'll go to the center and you will be evaluated for the first step, which is apheresis. So many of you have had a stem cell transplant and you remember that you had to have your cells collected, and that usually required 4 days of Neupogen, and then putting this catheter in your chest wall and then a couple of days of apheresis. So actually it's much easier than that. This is just 1 day of apheresis, and it's usually 70% of the time we can do it just with an IV. We don't have to put in a catheter in the chest wall. And that's actually really great because patients come in, they get 2 IVs put in, they sit in the machine, and 6 hours later, we've collected cells. This is usually all it takes, just 1 day of collection, and then you go home. That is a time that you're going to have your caregiver come bring you that day, just because you want to make sure you're not too tired. Everyone remembers sort of what apheresis kind of feel yucky during that time. But that's usually a not complicated procedure. Then you go home, and then it takes about 5 weeks for the cells to be made. And depending on where your myeloma is, some people have sort of a slow upticking myeloma, and they're kind of cool coasting for 5 weeks, and other people, not so much. And so those people may be getting a cycle of therapy, maybe carfilzomib or Cytoxan. You have to make sure you have therapy only until 2 weeks before the next step, which I'll talk about. So there will be a 2-week break between your last therapy and step 2 of this CAR T program. So step 2 is the actual whole CAR T enterprise that comes to you. So these cells get shipped off, I think, to New Jersey, and then they get come back, and that takes 5 weeks. The reason it takes 5 weeks is, of course, you got to make the cells, so you got to engineer them and do all this, new age stuff. But you also have to do quality control. Remember, these cells are being engineered, they're being genetically modified. So you have to make sure these cells are not going to just take off and grow on their own. And you have to make sure that there's no bacteria or virus or all the stuff that likes to grow in cultures, right? We're trying to grow these cells mimicking what it would do in a body. So you have to just make sure it's all clean. So that takes a lot of QC or quality control. And that's what takes some time, but that's all for your safety. So ultimately, these cells will be shipped back, and they'll be shipped back to the laboratory again at the center. So the step 2 also has to be done at the specialized center, even though that in between time, that 5 weeks, you may go back home and get some chemo from your local doctor. So during step 2, there's also a few parts. So the first is that you have to have 3 days of chemotherapy. This is what we call lymphodepleting chemotherapy, or LD chemo for short. And the purpose of this chemotherapy is to make room for these T cells. There are 2 drugs that you'll get with this. One of them is called cytoxan or cyclophosphamide. Almost everybody will have at least heard of this or gotten this drug already. It's a pretty small dose, so it's nothing like the high doses that people get. It's pretty well tolerated. And the other drug is called fludarabine, and that's a drug you will not have seen probably. And what that drug does, but that combination, is they actually kill the existing T cells that you have in your body. Why? Because these new T cells that are going to come in, they need that milieu. They need that environment where nothing else is fighting them for this space. And remember, these T cells that come in, they are your T cells, but they've been genetically modified to be a little bit different. Right now, they express this protein like a little lollipop on the surface, and they want to go and attack the myeloma. Well, that's not what they normally look like. So anything that's abnormal, your own T cells think, hey, you're not supposed to be here. So they might reject those T cells. So that's why we give these 3 days of chemotherapy, fludarabine, and cytoxan. It's very well tolerated, mild nausea at best. We do adjust it for kidney insufficiency, so we may lower the doses, and you can get this as an outpatient. So this will depend on the logistics of your own center. So like at our center, patients that live far away might just be admitted for this, but people that live, within a 30-mile radius may just come every day and go back home, sleep in their own bed. Or some people want to get an Airbnb because they hate being in the hospital, as they don't want to do it as little as possible or as much as possible. So this will really just depend on what structure, very similar to the transplant, where there's sort of different ways to approach it. Now, you get that chemotherapy for 3 days, and I'll just give you a sample schedule. So we usually do like a Wednesday, Thursday, Friday, and then there are 2 days of rest. So Saturday, Sunday rest. The reason we give that rest is we want all that chemotherapy to wash out before we're going to give you these cells. So you have Wednesday, Thursday, Friday rest, Wednesday, Thursday, Friday chemo, Saturday, Sunday rest. And then on Monday, those cells will be infused. Now, for right now, we are planning to do this as an inpatient, particularly for this ide-cel product, because we know that the majority of patients have the CRS, or the cytokine release syndrome, on day 1 usually. And that's right after the infusion. So it makes sense to admit them knowing that they're going to probably have a fever or something like that. So we're going to admit people. It may not be the same at your center, so just check with what their operating procedures are going to be. But generally, we admit the patient, they come in. We may admit them the night before just so we get everything squared away. And it's pretty anticlimactic. The cell therapy people will show up very similar to the transplant, and they'll check all the bags and then infuse the cells. You'll probably get a PICC line or something like that to infuse the cells in. And then you just hang out. And during the infusion, you may have some chills because it's a new product and it's been frozen and all that stuff, but generally well tolerated. It takes like 10 minutes. It's, like I said, really anticlimactic. And then after that, you'll be watched. That night may be the first time, or the next day may be the first time, that you'll have a fever. And at first, people may give you Tylenol. You may be seen by the doctor there. They may ask you some questions like, how are you thinking straight? Make you write your name or a sentence. And then if the Tylenol doesn't work, you may get tocilizumab, which is the antidote I was telling you about for the fever, cytokine release syndrome. Of course, it's, everyone's jumping on everything. So you may get antibiotics, you may get a chest x-ray, all the things that we would have done if you got a fever during the transplant. I will say that the chemo you get with this and the cells you get, it is, in my opinion, not as tough as the transplant. When people have gone through transplant, it's like their entire GI system just goes through a total whirlwind. This is really controllable nausea and it's gone pretty quickly. Fatigue is the main thing, but that's because you feel like you got the flu, which is a good thing actually. And so that's the main thing people complain about. For the next couple of days after the cells are infused, if you have the cytokine release syndrome and it resolves and you're good for 48 hours, you may be discharged depending on what your center does. At most, you'll stay there for 14 days, but I really doubt it for this particular FDA approved product. When you get discharged, one of the things we watch out for is, hey, do you have another fever? Or do you get confused and have neurotoxicity or low blood counts? Those are the three things that first 4 weeks after getting those cells, let's say you got it on like Monday, March 1st, through Monday, through the 28th of March, you're going to be watched at least twice a week in the outpatient setting, similar to like an outpatient transplant program or post-transplant follow-up program. And we'll ask you these questions and we'll see what your blood counts are doing. After the first 14 days, I'm okay giving things like Neupogen, which is growth factor. You may need transfusion, though it's pretty rare. And even after you go home after that first 28 days, we're in really good contact with your doctor because you may still need a little bit of Neupogen. You may need help with your blood counts here and there or with hydration. But usually we check those myeloma numbers before you go and see where you're at there. So that's sort of an overview of the first month. And after that, we kind of figure out what you need from what the side effects are. And then we watch and see how your disease does. And usually we check a bone marrow around the third month. And I always send for minimal residual disease because we know from the phase 1 trial, the prior trial, that those who had MRD negativity did better. And we'd like to inform our patients about what to expect during their post CAR T course. So Jenny, I don't know if that was a good or, I mean, if you want
Yeah,
to.
no, that was fantastic. You got through a lot of content in a very short period of time. Do you also want to talk about because I love how you went through the process, the caregiver
Yeah,
responsibility, like what's required by the caregiver,
yeah.
practically speaking?
This is really important. And I think one of the things that may differentiate where if people get bites versus CARs, caregiving for CAR T is very similar to what it was for transplant, meaning we need somebody to drive the person for that one apheresis day. Okay, that's fine. I mean, that's easier. But for the actual post CAR T infusion, we do need someone around for about 28 days. And it doesn't have to be the same person. With COVID, it was a little harder because we didn't want like 50 people going on. But now that gets better, it could be a schedule of people that, 1 week for this person, 1 week that person, mainly because if there's a fever, the person has to immediately go to the ER. Why? Because they may need this antidote. So very similar to fever after a transplant, you want to jump on that quickly for the
Yeah.
treatment for it. So it is a 28-day responsibility. I definitely think that the patients are healthier after CAR T, meaning they don't have as much GI stuff. So like the caregiver hands-on responsibility is not so much, but the being there is, they have to take off time from work or work from home or whatever. They physically have to be there.
Yeah, and just notice, are they acting funny or did they have a
Yes.
fever? Yeah. Yeah. Okay. So it's about 28 days.
Yes.
Okay. And is that including the time? So let's say you're in the hospital for 10 days. So it's just in the following 18 days
Yeah.
after
It's
that? Okay.
exactly 28 days after infusion of CAR T. So there's those couple days before with the chemo. And yes, it's nice for the caregiver to be able to drive them to chemo if necessary. But
Right. Right.
the where you could cut it off and say, okay, I'm going back to work, tell your employer, is the 28 days after the CAR T infusion.
Yeah. Okay. And then can you talk a little bit also about how centers are the specialty centers, because not everybody could administer this, right? So
Right.
do you want to talk about what you learned as a center and how, because there is a finder, if you, the website that you mentioned, abecma.com, does have a finder, and new facilities are coming on almost every day to be able to produce or to be able to administer this.
Yeah, very good question. So the, um, the website does show where you can go, but how that is determined is based on training. There's two aspects. There's contracts, there's money, and there's training. So the site has to be contracted to do it, and that means it's all good with the contract. I don't deal with all that. but their site staff has to be trained. So what did the company who makes this product do? They conducted these studies all over the US and globally so that each site had had experience doing it. So our site was like the second highest enroller, but Stanford was in the phase one, so they know how to do it. So
Mhm.
that they were the company was pretty smart about this, and all of us have to have training. So we have to have training because it's a very specific side effect profile, which requires a risk mitigation strategy or a REMS program, which we have to have the patient enroll in because it's enough of a risk for a couple of things. One, the CRS. Two, they're getting a genetically modified therapy. So we got to watch out, make sure nothing weird happens with those genes. We haven't ever seen any of this cause a secondary problem. However, it's something we do modify, we monitor the blood for. So it's really important for us to be able to monitor closely. And that's why this company that makes Abecma makes sure that we're trained and that we're enrolled in the REMS, that we pass a quiz, and that we're constantly, everyone who comes in that works at our site has to get trained on this. And that's for your safety because you don't want people that have never given this therapy before to just be giving it. There are too many steps. I mean, the steps I told you was just, 1 through 10, but there's a bajillion, one sub B step, in there, a lot of chain of custody, a lot of logistics. And this is for your safety. So I would say if you can find now, Abecma.com does have the sites that are approved. And then if you are near one of those sites, then you can contact that site to see. Usually they have an intake navigator person who will funnel you to the right myeloma doctor. And you might even just set up to do a Zoom, for example. And say, okay, I'm in second line or third line therapy right now. I want to just get in touch with you. So when I need this, I'm on your radar, if that makes sense.
Yeah, and then I mean, you talked about lines of therapy, and just as an example, like I did, my tandem transplants, some induction therapy, tandem transplants, a year of consolidation therapy, and then another year of maintenance, and that was like one line of therapy.
Yes.
So it seems like not that much, I mean, when you count it, but it was a lot of therapy.
That's a lot.
Yeah, yeah. And so, do patients ask their current doctor, like, if I wanted to, my facility, let's say I'm a local general oncology center, they don't have this available, but I have somewhere that's close by. How do I logistically do that? Do I just call the other center and say, I want a second opinion, I want to get screened? Do I have my doctor call that facility for me, or
Right.
like, how does that work?
Yeah, there's a variety of ways it can work, and it really depends on the center you're at. So let's say you're at a community cancer center, you get your infusions there. Usually your doctor will know someone at the near center because they
Mhm.
do a lot of referral. If you got transplant, for sure, that your transplant doc is the person to go to. That's already
Mhm.
like in the bag. And so if you're already kind of peripherally followed by your transplant doc, which I do follow a lot of my patients, that's good. Now, if you're sort of de novo, have never been to one of these centers, first thing would be to ask your local doctor, say, hey, do you know someone there I can get in? But every single one of these centers should have a website. They should have a way to call in. I mean,
Mhm.
it's all different, but there's a way to get in there. I
Yeah, there's
really
like a new patient number or
new
something you
patient
can
number,
call.
yeah.
Uh-huh.
But
Mhm.
your oncologist, most oncologists, they are really good about, hey, I'll put in a referral, I'll follow
Mhm.
up, that kind of thing. And either I think having a two-pronged approach where you do your part and you ask your
Yeah,
oncologist to
yeah.
do her or
Because you
his
don't have
part.
to wait either, you
Yeah,
know?
you don't want to wait.
Yeah.
That's why I think, Jenny, what you're saying is so important about being proactive because, like I said, if you've relapsed once or twice and you're still not eligible for a Abecma, right? It's like after the fifth line or fourth line. You still want to get in and meet someone that might be able to be a specialty center because maybe 2 years from now you will need this. And it's good
Mhm.
to just have a Zoom and say, hey, I'm here. I'd like to just plug in with you. And you have a medical record number. We have our insurance, like everything's all squared. And then
Right.
when it comes time, it's not a rush appointment. You just slide right into a follow-up slot.
Okay, that the practical aspects of this I think are equally as important as the science because sometimes it's just,
Yeah,
it's just practical logistics.
absolutely.
So, okay, one other question, and then let me just intersperse my questions with questions in the Q&A.
Sure.
So we get to those. Okay, Dave is asking, do you have any thoughts on the KarMMa-4 study for newly diagnosed myeloma? So the KarMMa studies, if people don't know, are what got this drug approved, Abecma approved, and those are, they like when they name clinical trials, maybe you can just kind of comment on how that's done.
By some marketing person at the pharma person. No. Yeah, exactly. They figure out some cute what is thing's called?
Yeah, mnemonic or something.
Yeah. So to answer the question about the Karma 4 study, I can because I'm one of the lead investigators. The Karma 4 study is using the same Abecma product, ide-cel, and giving it to patients with high risk multiple myeloma. So if you have revised ISS stage 3 disease, now that's a very specific population. That
Mhm.
means that your beta 2 microglobulin was greater than 5.5, and you either have high risk fish, and I won't go into all the fish details because it's confusing, but there's 3 fish, or you have an elevated LDH. This is all a diagnosis. If you have those, then you can go on the Karma 4 study. And what that is you get your induction 4 cycles of chemotherapy. And somewhere in there, that between the third and fourth cycle, we collect cells to be manufactured to become CAR T. And after that fourth cycle, you actually get CAR T. You don't go to transplant. You just get CAR T to see if that will do a better job of keeping these patients in remission, because we're not doing a good enough job as it is. So for anybody with newly diagnosed myeloma, I highly recommend that you ask your doctor to refer you to a specialist just for an initial Zoom. You don't have to do anything that the specialist recommends. They may or may not recommend the same thing. Two heads are always better than one. We have a great
Mhm.
relationship with all of our referring docs. It's always good we text back and forth about, okay, should we do this or that? It's better to just get more neurons on board to
Yeah,
try
I
to
agree.
address this. And maybe you qualify for this. So that would be one thing.
Okay, I agree. You should always get a second opinion. Okay, Susie's also asking a question that I think is an interesting question about follow up for CAR T. Like, what therapy is available after CAR T, assuming it's successful for a year, and what happens next? But I want to open this up for a broader discussion about maintenance therapy after CAR T, because in the clinical trials, obviously, they want to see if it works and how long it works. So it's a one and done therapy, and then you don't do anything afterwards. But now that it's approved, I don't know. Do people do maintenance therapy? I guess it's not really off label because you might do, I don't know. How does that work?
Yeah, that's a great question, and something that is being explored in other karma studies. Not don't forget, there's karma
Mhm.
1 through 7.
Yeah,
Karma
yeah,
7.
yeah.
So some of these studies are looking at giving these T cells and then afterwards doing maintenance therapy to prolong the duration of response in
Mhm.
various lines of treatment. So not just the really late line, but
Mhm.
completely sticking to the label, completely sticking to what the data is right now for safety and for efficacy. This would be a one and done CAR T therapy, no maintenance. And the
Mhm.
reason for that is that it wasn't studied that. We don't know what the safety will be. We don't know what the blood
Right.
counts will be. We don't know what the IMID or whatever maintenance you would do to the T cells. And we don't want to rock the boat. We want to tell patients you can expect this. This is what the data shows. This is what it is. And most people will have already gotten maintenance lenalidomide or have been exposed to pomalidomide or some one of these drugs that's available for maintenance. People will have already gotten that. And remember that a lot of other drugs, for example, cytokine or steroids, they actually kill cells. Well, you don't want to kill the T cells. So there's, I would not put my stamp of approval on maintenance now outside of a clinical trial.
Okay, good to know. Okay, CM is asking, what are the changes on finding a generic or off-the-shelf kind of CAR T treatment? He's in Holland, so kind of wondering. I mean, that whole landscape is evolving too, right? But
Right. So just a word about the allogeneic or off-the-shelf CAR T cell. So as I already explained, this FDA-approved product is something that you take from the patient himself or herself and engineer it. Now there's another product that's being developed by a couple of different companies. That's an off-the-shelf CAR T product, which is that they take random donors, they take their cells and engineer them to do 2 things. One, to kill the myeloma, good, but also so that they don't kill anything else in the recipient body. Remember, if you took someone else's T cells and put them in your body, those T cells would have a party and they would attack the person's body. That's why we are very careful about allogeneic transplant. We match people. So people have been very smart to figure out how to get these T cells to not attack the patient himself or herself, but rather to only attack the myeloma. And what they've done is they've taken out the T cell receptor and replaced it with the CAR T protein, the lollipop that sticks on the surface of the cell. And that's really smart. But this is going to take a lot of science, a lot of time, and a lot of clinical trial data to understand if it's going to be on par with what we have from the autologous CAR T cell studies. And I think there will be a place for it. Not everybody can do the collection, not everybody makes enough cells. And that's what the landscape will be. There will be a place. There's enough myeloma to go around. There will be a place for that, but we don't know how good it's going to be and how safe it's going to be just yet.
Yeah, yeah. That'll be interesting to see how that works, but between your own cells and the donor cells, that's fascinating. Dave's asking another question. Is there a correlation between how high of a disease burden you have at the time you go into CAR T treatment and the cytokine release?
We thought there would be, but not necessarily.
Yeah, I think that's super interesting because it's also not the level of BCMA that you have either, right?
Yes,
Like,
it doesn't matter. Like we
yeah,
apparently even low level BCMA responds to CAR T. So it's a mystery.
yeah. Okay. George is asking, is there a difference between, I would think of this in the context of CAR T, is there a difference between treating patients with extramedullary plasmacytomas? Well, he's asking between bone and non-bone, and do you treat these patients differently? So I guess you can answer the question in general, and then as it relates to CAR T.
Yeah, so there is some subgroup analysis with extramedullary plasmacytomas, and people did respond to CAR T. I will say that what we call EMD, extramedullary disease, is a different beast, and it's more aggressive. And so we love to be aggressive with those people. We have to be. So I cannot tell you which therapy is the best for that at all yet. Hopefully one day we would be able to, but they haven't enrolled people like just done an EMD cohort. Like that would have been useful, right? So, but they didn't
Yeah,
do that.
yeah.
I have had people who've responded to CAR T with EMD.
Mm-hmm. Interesting. Okay. And KT is asking, how many times can a patient receive CAR T therapy? So,
Yeah.
and do you want
Sorry.
to talk about boosters or
Yeah.
whatever too?
Yeah. So, really great question. This is my opinion. Once you've gotten one CAR T cell therapy, you should not get the same one again, because there's no data that I've seen that shows that retreatment with the same product works.
Mhm.
However, there is some data, and I even have some anecdotal experience where people have progressed after a certain CAR T, or even maybe like they progressed after Blenrep or something that's BCMA directed, and they respond to another CAR T or another BCMA directed therapy. So you may get a
Mm.
CAR and then a BiTE, or you may get a CAR and then a different CAR, or Blenrep and then a CAR. So those are interchange. Like you can mix and match with that, but retreating with the same CAR T, I do not think is probably the way to go.
Yeah. So yeah, with the same target for sure. Okay. Amy's asking, do you lose all your immunity like you do with a stem cell transplant because you have low dose chemotherapy? And then what's the impact on your immune system with just the CAR T? Like what you're doing these combined, I mean, it's lower dose and everything for the chemo, but interesting question.
Yeah. So interestingly, the immune there is a profound immune effect on this, and the reason for that is that these T cells are killing every B cell in sight. So the B cells are the ones that produce antibodies, as you know, because of the plant's plasma cells produce these proteins, and they do such a great job that you may have what we call hypogammaglobulinemia or low IgG levels, and
Mhm.
maybe you will be getting IVIG. We did that a lot in the study because people just like had such a great response, they have no B cells left. And this happens also with the bispecific T cell engagers, also known as the BiTEs.
Mhm.
So that is a major effect. So it's not just the fludarabine and the cytoxine, which is really not so much, it's the actual effect of the T cells that are there to kill the B cells that have done a good job. And
Mhm.
so you will have low immune system from that. And you may not respond maybe like to COVID vaccine. We don't know. We're looking at that right now because you
Mhm.
don't have the antibody means to respond. That being said, I just want to say one more thing about this COVID vaccine business because it's like
Yeah, yeah.
very confusing.
Right.
A lot of patients we're testing, everybody's testing them for the COVID antibody after they've been vaccinated. So if you've been vaccinated twice, the two sets, and you wait 4 weeks, you can get this antibody test done. Now, if the antibody test is positive, great. Okay, awesome. You're probably okay. If the antibody test is negative, what that means is that your antibodies aren't being made. And those are things that are made by B cells, which is the same group of cells that your myeloma comes from. So it probably means your myeloma doctor is doing a great job killing these B cells, but it doesn't measure what your T cells are doing. And we know that both the Moderna and the Pfizer vaccines, they also rely on T cells, which is a different group of cells to kill in a different mechanism, not by producing antibodies, but really engaging cell to cell, just like T cells do
Mhm.
with this to kill the target COVID. Now, we can't measure that with the commercially available test. There are some experimental assays to do that. So what I'm saying the whole reason I'm saying this is that yes, there are effects on immunity, and yes, your COVID antibody test may end up being negative. That's not to say you're super unprotected. You may still have T cell protection, but we can't tell you that because we don't have a commercially available test to do that. And did I cause confusion? I don't want to.
No, that makes a lot of sense because you get it, the antibody test, and you're like, oh, well, I didn't develop any antibodies. So now I have to be, like, well, you probably do want to take more
You
still
want to take precautions.
just in case. But what you're saying is you might actually have T cell protection
Right.
by getting the vaccine. So don't not get the vaccine, right?
You should definitely get the vaccine. And I actually, because then I had a patient yesterday saying, okay, should I get a different vaccine? I'm not sure that's going to work because all I found out from the fact that this person did have COVID, did not make COVID antibodies, is that his B cells are not working, which is not going to change no matter what vaccine he gets. So,
okay, I have a question on that topic. So if you're trying to figure out who's going to respond to CAR T treatment, who's going to get into this complete response like you talked about earlier, because I know some patients who are 3 years out from these karma trials and have not relapsed because they got into that deeper response. So, like, how do you test for that? That would be nice to be able to say, well, you're likely to get a 3-month remission or something with CAR T, and it's kind of a big deal to do, and it's very expensive. And so, but is that something that is available or will be available?
So this is such a great question, and the person who wants to know this the most is the insurance company because they would love to be able to predict where their $400,000 is going to go. So we are actually, first of all, the short answer is no, we don't know, and that's why anybody who was involved in any of these clinical trials knows that they had like a bajillion tubes of blood taken because wanted to predict. We are actually trying to, hopefully this gets funded, launch a project between UCSF and City of Hope where we are going to take peripheral blood, so just the blood that people get or part of the apheresis product. I'd rather do it just from the blood people give and look at that from a protein standpoint and a DNA standpoint to say, hey, correlate that later. Did this person respond? Yes or no? Okay, they're in this category. Do they respond? Yes or no? Okay. And see if we could predict this. There's so many ways you could do this, right? Proteins, RNA, DNA. See if we could find a signature, because if we found that you're likely to respond based on a blood draw, well, I wouldn't put someone through a whole CAR T thing unless they were likely to respond, right? So these are the things we have to ask because, we're asking a lot of things from patients and maybe they should get a different therapy instead. So we don't have a way to know that yet, but I'd love to be able to find that out, and that will take years of research to do.
Okay, Michelle's asking, do you feel that this is a good therapy for someone who's had an allo stem cell transplant? Interesting question. Who's also currently refractory?
Yeah, I don't think there's any reason why an allo patient cannot get this, because if you've progressed after allo transplant, the graft is useless, right? So you basically have your own cells, and I think that's fine.
And then someone else is also asking, what's the efficacy that you saw in all the karma trials for patients with high risk genetics? And I know patients who have had all these lines of therapy, 6 average lines of therapy or whatnot, probably have some kind of high risk genetics, which is why they're relapsing so many times. But is there any data on that?
Yeah, there's some subgroup analysis. It was a little bit less. Everything high risk, you can just -5 points, like it was less. There are some people that responded, but those people tend to stay in response for less time.
Okay. And do you know of any Canadian CAR T hospitals or programs? Like, is it open at Princess Margaret or other places? Are you aware of that?
We had Canadian sites on the karma trial, so I know that there are sites, and I just don't know, I forget which one. It was an early investigator call for me, so I didn't know all the sites.
Okay. Well, that's probably on the list, I would think. Okay. So someone's asking, what do you think of other, I guess, competitors when you're thinking about these BCMA targets? So Blenrep is one as an antibody drug conjugate. You have a whole host of bispecific antibodies coming out. When would you use the CAR T, Blenrep versus the bispecific antibodies? And and let's see. Are there any other BCMA targeting medication? Well, there are a bajillion, but it seems like.
Come to one of my talks. But that's a great question. And the reason it's good is because not all myeloma patients are the same, right? Not everybody should do the same treatment. So my personal feeling, and this is just my algorithm, and not everybody's going to agree with me. So my personal feeling is you have relapsed refractory myeloma, you've had 4 prior lines of therapy. If you can make it to CAR T, you should go there. Why? Because there's a chance you're going to be in this duration of response, maybe for a year, and you don't have to have any therapy. Wouldn't that be
Right.
great?
No,
Go to your
no
kid's
treatment.
wedding and do all the things that you want to do. Okay, maybe. But then the question is, okay, fine, that's my first choice. But the next algorithm, is it logistically feasible? So can this person come to get apheresis, go back home, wait the 5 weeks? Can they come back? Can they make it 28 days in San Francisco? These are the, yes, they can.
Mhm.
Okay, they go to CAR T. No, they can't. Can they come one time? Can they come 2 times? Two? Good. They can maybe get a bispecific, which requires hospitalization for maybe the first or second doses. Now, that's on trial only if there's a slot. And that's fine. Can they not? Okay, then they should get BlendRep, which they can get off the shelf in the community, and I can manage them remotely with their doc. So there's all sorts of sort of algorithms that I think about. Of course, the patient is you can't predict, just you have to know who the patient is, but that's kind of how I think about things.
Yeah, and their situation. Okay, that brought up a question when you were going through that. So you have to stay near the center for the 28 days?
Yes.
Like, okay. So that's like something you have to think about for
Yeah.
logistics.
So
So it's
someone,
not like you get out of the hospital after that 10 days or 14 days or whatever, and you go home and you're like, okay, my caregiver's watching me. But no, you have to stay near the center.
and
Okay.
the reason for that is that the antidote for the CAR T fever is probably only going to be available at the center, right? We have to have
Yeah.
on formulary ready to go. So you want to be, or if they have neurotoxicity, you need to know, you need a specialist.
Yeah, you do, because like for the CRS, you would use the tocilizumab, but that actually doesn't work for the neurotoxicity.
Right. Yeah,
And you
exactly.
use the steroids for that. And so
Yeah.
you go to a general center and they're like, we don't know, we're going to give you tocilizumab, but it's neurotoxicity.
Right.
And yeah, then that's not good. So,
Right. And I
um,
will say,
yeah.
just to add,
No, go ahead.
my understanding is that for Abecma and even like lymphoma, Kymera, and all that, there's some supplement or assistance offered by the company to help you to stay near the center for that time. So I would really go on the website. Like I said, I don't work for these people, so I don't know. But
Right.
my social worker really goes deep into all this stuff because we want access. We want patients to
Yeah.
have access to what they need.
I believe it does cover all travel that's associated. It doesn't cover, from what I understand, it does not cover screening. So, like, let's say you want to come to your
Right.
DCSF and get screened, but I believe there is a program, I think through the LLS, that actually will cover. I'll double check on who provides it, but there is travel coverage for $500 to cover for screening that's not covered
Company.
by the company. Yeah. So if you want to go get screened and, make sure you can have this treatment, there is coverage available for that. Okay, so someone's also asking, like a small percentage of patients enrolled in the study weren't able to get the CAR T. So like what happens? When does it not work during the manufacturing process?
Yeah, great
Or
question.
why?
Yeah, it depends on the product. So I'll just say for the Karma study, Karma 1, excuse me, I think they only had one manufacturing failure and they were able to remanufacture because when they collect the cells initially, they won't take all of the bags that they collect and expand them. They'll only expand some of them. And so if that for some reason is a failure, they have backup cells and they'll expand those. That not all patients, but a lot of patients. So that person eventually got treated. Now there are people who also did not make it to the CAR T cell. Why? Because they got too sick between the collection
They couldn't
and
wait.
the infusion. Now this is where I think we can do a better job with the FDA-approved drug because we can predict. Like, I'm not going to send my patient who's like blowing up myeloma. I'm not going to wait for that to refer him or her to CAR T. I'm going to go further, before that. So in most cases, you have some time with the myeloma to not let it get to that point. We, as the clinical trial people, we were just putting people on there, on death's door, honestly. And so
Right.
that is what happened.
Right. And I mean, yeah, I had a friend who was on 18 prior lines of therapy. So that's
Yeah.
like an insane amount. And yeah, if you can do it right after the 4, then
Right.
you're more likely, you don't have to wait for the 6 or the 10 or
Right.
whatever. That's just people who qualified. Okay. Vicky was just re-asking what the chemo was, but that was cyclophosphamide and fludarabine, right?
Correct.
Okay. Um, Okay, Eloise is asking a very good question that I think will come up a lot with patients. What factors will you decide to do a second transplant versus CAR T, assuming, same
Yeah,
lines of therapy?
this is a fantastic question. So, and I haven't decided yet. um, I think what I'm gonna do, I think, is I would. And by the way, I'm a recovering transplanter, so I love myself some transplant. So, I think what I would do is potentially do the CAR T first, but have the second transplant as a backup because those cells are there, and I don't want to give the melphalan if that doesn't work, because then now I've given this toxic chemotherapy that might affect the T cell population that the patient gives for the collection. So I'm just trying to like immunologically think what's
Mm-hmm.
better first. So I probably would do the CAR T first, see if I can get a year of that, but be ready that the patient may need a transplant because the CAR T is not that toxic. I mean, from like a traditional
Hmm.
chemotherapy standpoint.
Well, you always know you can go to transplant, right?
Yes.
And then, like, is there a window of time if you get a second transplant that you have to wait to get
Yes.
CAR T therapy?
Okay, so that was true on the clinical trials. It was like 3 months or 6 months. You couldn't get a recent auto, but I mean,
Oh,
that's really because of the tolerance. Well, that's because the drug companies don't want something bad from the first transplant
right,
affecting their
right.
data.
Uh-huh.
I don't really think that's medically necessary. But what I do think is having gotten um, the melphalan, okay, sorry, I said that wrong. So you could do it either way because if you think about it, when you get melphalan and you get that second transplant, you then reinfuse this brand new immune system. So that
Right.
actually, so
Oh,
so
interesting.
I realized,
Lots of ways to think about this. Like,
yeah,
yeah, that's why you need a specialist.
yeah. Even I'm thinking, so let me just answer that question again. If the person had had like a ridiculously long amount of time after the first transplant, which a lot of people do, like 4 years,
Right.
then I would
Hmm.
say, hey, do you want to do the transplant again? You might get 2 years out of it, and from CAR T, we know you might get 1 year. I don't know. You decide. These are
Hmm.
all numbers. I don't have a crystal ball. This is just stats. And
Right.
patients say, I don't mind. I'll do a Mel 140 transplant, so less Mel, and I might get
Hmm.
a couple of years and I was fine and I don't have to collect cells again, so I'll do that. So that's fine.
Hmm.
Or they might say, you know what, I hated that transplant. I don't want to do it. Let me try this first and then do that. Because even though we give the melphalan, we give a whole brand new set of stem cells. So those cells are okay.
Right. Yeah. Okay, that makes sense. There's a lot of different, and that goes back to personal goals too, right?
Yes,
Like how was that for you? Some people are really slammed by transplant, and some people not so
yep,
much. It was not that hard.
exactly.
So yeah. Okay, KT is asking, when you talked about the data and the complete response, what percentage of patients, or how many patients have exceeded that 1 year response rate, I guess? Or what percentage of patients got into that complete response?
Oh my gosh, I don't remember the data exactly. I think it was something like 30%, but I'm kind of lying.
Okay. No, that's
Sorry.
okay. I would have to go look it up.
Yeah, I'm sorry. I didn't bring the slide. So
Okay. And then how long, I mean, we're all very excited, and myeloma patients are super impatient, and something new gets approved like this, and we're all very excited about it. And then we're like, well, okay, now when is it going to be approved for newly diagnosed patients?
oh God.
So
Yeah.
how long do you think it'll take to go through the trials for newly diagnosed, and just bringing it up earlier in the clinic? But multiple years, I would expect, but
So I think what's going to happen, this, the good news is we're doing awesome for newly diagnosed myeloma patients. The bad news is we're doing awesome for newly diagnosed myeloma patients.
yeah.
So what I think is going to happen is probably this CAR T therapy will not just be approved in the fifth line, but these ongoing clinical trials will allow it to be approved in the second to third line, meaning that after your first progression, you can get CAR T. So there are already trials ongoing for that, and they're almost completely enrolled. Okay, if that gets approved, and that's even going to be tough because that sometimes requires a randomization. Some of these have like Karma 3, I think is randomized. So fine. If that gets approved, then the big hurdle is upfront. And I'm not sure that it necessarily should be upfront. I don't know yet. But to prove that, you would have to have a randomized control trial of people getting upfront CAR T versus traditional chemotherapy, transplant, maintenance, which alone, depending on what chemotherapy you get, can give you 4 to 5 years of
Hmm.
stable disease. And
Hmm.
how are you going to get somebody to fund a trial like that where they're going to be waiting at least 5 years to get data, right? Who's going to do that? So it's a tough clinical trial hurdle to climb. And there's going to have to be some innovative ways of doing this. I'm not sure yet because I actually think that I think, people like to compare transplant and CAR T. And it's only because the word cell is used. There are totally different therapies. Transplant
Mhm,
is
mhm.
old school Brillo pad chemotherapy, and it works. It does have this ability to kill myeloma cells that are not so easily findable by traditional Velcade Rev Dex. And
Yeah,
CAR T is a totally different. It's an immunotherapy. So it's like you're comparing, one sister versus another. It's just like different people.
yeah.
So I don't know that we'll necessarily make it up front. What I do think is going to be the next thing is taking patients up front, getting induction chemotherapy, transplant, and then some people get randomized to get a CAR T. Some people get randomized to get just maintenance, and then there's maintenance after for every.
Oh,
I think that'll
that's
be like a 1-2. That'll be a design.
that'll be really nice because then, yeah, you don't have to think about doing maintenance therapy for years on end
Oh no, no. I
or
think
just
they'll
like
make you. Well,
they'll
I
make
think
you do maintenance.
they'll make you do maintenance because the FDA always wants you to know that you
Yeah,
didn't deprive a patient of
right.
something that is FDA approved,
Okay.
right?
Makes sense.
So
Okay, we're running out of time, but
yes,
I'm
I
going
know.
to
I
ask
gotta go
two
too.
more quick questions. I'm sorry. So do you lose your hair during that CAR T induction therapy?
No,
Yeah, I didn't think you did because it's very much more mild than the melphalan.
right.
Okay, last question. After CAR T, what should patients consider? Yeah, and then where do you see things going to maintain responses for CAR T?
I think probably some maintenance IMID might do this, but after CAR T failure, another clinical trial always. And if you're starting to progress a little bit, like not clinically, maybe another immunotherapy trial to boost up whatever T cells may be there. And we're doing some of those trials at UCSF. So that kind of thing. Most importantly, to just have really good communication with the referring doc and between the community doc and the referring doc, because the more options you have, the more you feel empowered to make the decision that's right for you.
Hmm. Okay. Well, this has been an amazing, amazing discussion today, and that's so informative. There's so much to learn from just the practical aspects of the therapy. So thank you so much. Super meaningful for patients.
Great,
You answered a
great.
ton of questions. So thank
Thank
you.
you so much for having me, Jenny. I love what you're doing, and you're the most. You're like the fifth vital sign here about patient education. So I just love that you educate us, and it's the most important thing we can do to empower patients is education. So I'm
Yeah.
very privileged to be a part of your team.
Well, thank you so
Yeah,
much for being so generous with your time.
Great. I'm gonna leave because I got to go to another meeting.
no
Yeah.
problem. Thank
Okay.
you.
Zooming away. Okay. Bye.
We'll just
Bye.
close
Thank you.
out here with a couple of announcements. That was amazing. I learned so much, and I love just the practical way that she approached that.
Mhm.
Our next meeting will be on Friday, June 4th, 2021. Obviously, I don't know why I include the year, at 1 p.m. Eastern. And we're going to be talking about the Skyline genetic testing for myeloma with Patricia Murphy. So don't miss that. And then we have various upcoming events because we're short on time. I'll just let you know that you can access these events by going to www.myelomacrowd.org/events, and that this link will be included in your follow-up email that's sent 24 to 48 hours after this event concludes. Thanks to our sponsors, Bristol Myers Squibb, Amgen Oncology, Karyopharm Therapeutics, AbbVie, Adaptive Biotechnologies, Genentech, Oncopeptides, and Takeda Oncology. And thank you for helping us build the strong myeloma crowd community. Like Dr. Shah said, it's the best thing you can do for yourself is being here, educating and empowering yourself. So thank you. We appreciate you and hope that you have a great rest of your day.
Bye, everybody.